This week's topic is bioarchaeology, or the archaeology of human remains. And so I want to begin with a quick content warning. This is to acknowledge that for many people, for many cultures, there are concerns, very valid ones, around the display of human skeletal materials, of grave sites. So I just wanted to give you a heads up. The content that is presented in this PowerPoint can also be learned by just examining your text, but just note that the images that I've used here are those that I either have explicit permission from community members or are those that have open permissions for educational purposes assigned to them. And so for the exam and for the assignment for this topic or the lab exercise for this topic, you won't be required to look explicitly at images of human remains. Instead, we'll have questions regarding the methods we use for analyzing human skeletal remains in archaeological contexts. So what is bioarchaeology? I love this basic definition here, which is the analysis and study of human skeletal material in order to understand the broader implications of behavior, to understand broader trends in human populations in the past. So this means it's multidisciplinary. So many bioarchaeologists will have a very strong training in biological anthropology, of course, but the other disciplines are also very critical and valuable. Of course, geology can be beneficial because of understanding the context of the remains themselves. Comparative anatomy, comparative zoology from biology is also useful. And then chemistry, in terms of doing analysis on the remains themselves, is also a critical subdiscipline. So bioarchaeology is a very multidisciplinary subdiscipline of archaeology proper. I like talking about this topic at this point in the term because it allows us to revisit one of the core concepts for this course, which is ethics and ethical approaches. As my content warning suggests, the study of human skeletal remains is an ethically difficult subject. This is because many groups, including many Indigenous groups, do not want human skeletal materials studied. They have very valid reasons, again, for doing this, not just kind of cultural, spiritual beliefs regarding ancestors and remains and the dead and the afterlife, but also because of the kind of dark history of anthropology and how the remains of Indigenous communities around the world were used to further our scientific endeavors. Okay, so what that means is in a contemporary context, ethics has to be first and foremost, right? Who are we studying? Why are we studying them? What's the value? And what are some of the risks and concerns around this? So a lot of cooperation and collaboration is necessary. Here in Canada, the excavation of human material is legislated both by provincial and federal legislation, but you have to keep in mind that, as in most archaeological work, that it's not just archaeological legislation that's important, but we have to think about other forms of legislation that might be relevant. So things like the criminal code, we have to make sure that we're not dealing with any remains that are contemporary or associated with an ongoing or could potentially lead to an ongoing criminal investigation. The Public Health Act kicks in whenever you're dealing with the remains of individuals. We have the cemeteries legislation, so there's a whole bunch of other things. So you have to be aware of what the legalities are of the excavation work you want to do. In the case of accidental discovery, the standard practice is to, of course, halt the work, protecting the remains, and then contact local authorities, including the police and RCMP, but also, I'd say, members of the archaeology branch, members of any Indigenous communities, to work together to find the best course forward. Another key part of the excavation of human remains is not just the excavation, but the repatriation, that many museums and universities across the world have human skeletal material and other objects of sacred cultural significance stored in their collections. So here in Canada, we follow, generally speaking, the policies of NAGPRA regarding the repatriation of human skeletal materials, again, with the goals of being very collaborative, very cooperative, but also ethical, transparent, and respectful. Parks Canada actually has some really great legislation relating to not just bioarchaeology Canada, but archaeology more broadly. It's probably the closest to a federal framework that we have. It's really excellent. And then, of course, here in Alberta, we have the Alberta Historical Resources Act. We've already talked about the ancient one, of course, which is one of the most infamous case studies relating to ethics and anthropological practice and research regarding human remains. So please do revisit our previous notes on this case study and consider this a bit more deeply now that we're further along in the term. In cases where we are able to excavate human remains, we have to kind of step back a few steps and think about some other factors that we have to consider. First and foremost, that one of the challenges we have with identifying skeletal remains in general is just identifying whether or not they are bone. In many burial environments, bone can replicate other materials, including stone and wood and other organics. So we need to be able to identify something positively as bone, and then we need to identify it as human. And this is where, again, that comparative zoology, comparative anatomy courses, comparative osteology, all of that training becomes really critical so that you can definitively determine whether or not the material you're dealing with is A, skeletal, and B, human. Then I mentioned, of course, the notification procedures may kick in. You need to know not only when to dig them, when it's appropriate, when you have the right permissions in place, but also what tools to use. Bone being an organic material is very different, again, in terms of how it preserves and the environments in which it preserves. So we must consider these factors. Generally speaking, we don't use things like metal tools because it can be very damaging. We tend to use things like soft brushes, wooden tools, plastic tools. So it's a whole bunch of consideration. Of course, how we handle them is impacted by some of our plans. So for example, if we want to do sampling for genetic analyses, we have to think about how we ourselves are constantly shedding our own genetic materials, so we can be a source of major contamination. So we have to think about that. I always emphasize that you want to have an expert on your team, a bioarchaeologist or a human osteologist or a physical anthropologist, someone who has that advanced training in identifying, handling, analyzing, and processing human skeletal material. And just to note that typically speaking, we do treat human remains as a feature, and this is why it's bioarchaeology, is that context that the human remains are found is really critical in terms of our analyses. So the next few slides, I'm going to show you some images from a site I worked on and some human skeletal remains that we accidentally came across. So feel free to skip ahead. So I use this picture here because I want to illustrate just how challenging it can be to identify bone and human skeletal materials in context in archaeological sites. So this is actually part of a cranium here. So you can see kind of the outline of the cranial vault here. A bunch of the bones of the skull have been already pre-removed. They were very thin, very flat. The skull itself was kind of compressed. You can see a couple of teeth that have been knocked out of articulation here. Next slide, you can see that the postcranial skeleton, or all the materials below the cranium, isn't much better. So we have part of a mandible here. We have some ribs through here. This is part of an arm. So we have a humerus here, radius and ulna fragments. Other parts of the skeleton had been previously removed, including some of the lower limb bones. So we have a lot of signs of disturbance. Preservation's an issue. And you can see here that this is why having an expert on your team who can identify fragmentary human skeletal material and very fragile material. So this stuff, if we touched it with anything more than like a soft brush, it would just basically fragment. So it became really critical that we knew how to approach it properly, responsibly, and respectfully. I'll also note that we ended up excavating the cranium en masse, which meant that we removed all of the sediment and all of the skeletal materials in a block. And then it wasn't until in the lab that the sediment was finally removed and separated from the very fragmentary cranium of this individual. I've already talked about the importance of documentation. So here's the form that we use for a burial record. You can see that there's a lot of details that need to be recorded, and how we figure out some of these attributes of the skeleton, such as sex and age estimation, I'll talk about subsequently here. Of course, mapping is really critical. So here is a reconstructed map of that individual that I showed you. So we did some piece plotting. Again, this is a highly stylized map, but this is based on the actual provenience of all the skeletal finds that we made, the dating samples we took, and also the rocks that were present in the sediment that impacted our excavation. So as bioarchaeologists, the context is just as critical as the skeletal remains, although I'll spend some time talking about that. I just want to mention that, of course, we have to think about where do we find human remains? Is it an accidental burial? Is it an intentional burial? Again, we consider it a feature because it includes not just the skeletal remains themselves, but all the artifacts, ecofacts, and other aspects that are associated with it. We can have single individuals, multiple individuals. We also bury non-human individuals like pets, so we have to consider that. We have to then think about the terminology that we use. What actually is a cemetery versus a burial? We have to think about what happens when we find commingled remains and some of the taphonomic processes or site formation processes that contributed to them being commingled. Again, was this part of the culture? Was this how human remains were handled and treated and interred? Or was this a post-depositional factor that influenced this? So again, context as everything, and we've talked about that so much that a lot of our interpretations of human skeletal material are strengthened and enhanced by our ability to analyze and interpret the context in which they're found, including their association with other forms of material culture. So preservation and treatment are also other critical factors. We have to consider not just what we're doing in the field, but how we're going to store and transport the materials, what forms of analysis are going to take. Is there any special testing we need to do? We have to consider risks of things like contamination. And then we have to think about preserving those remains after the fact, particularly when we're looking at repatriating them back to the site, is what is the best way to mitigate the amount of damage that's been done as we've removed them from their burial environment, their burial context. Okay, so those of you who take classes like Anth206, our Introduction to Biological Anthropology, will learn a lot more about this, but I did want to touch upon some of the briefly some of the methods that we use to analyze human remains, skeletal remains. So we can do sex estimation. We can look for differences in the skeleton that result basically from the onset of puberty. These are secondary sexual characteristics. We see changes in the skeleton associated with puberty that are usually meant, particularly in female skeletons, biological female skeletons, to facilitate pregnancy, labor, and delivery. So we see a lot of changes to the shape and size of the pelvis. Because we are a sexually dimorphic species, we do see some other traits that are just simply the basis of, again, male versus female. However, we do estimate sex using kind of a 5-sex scale. So we have male, probable male, ambiguous, probable female, female. And this is because we have clinal variation between these traits in males and females. Oh, so I have some here. I've also linked an excellent video that summarizes how sex estimation is conducted using the human skeleton here for you. Again, summarizing some of the key traits that are used in the cranium or the skull, and also, so here's some more, and then also those in the pelvis. Just note that sex estimation using the pelvis is far more reliable than using the cranium or any of the other bones in the skeleton. And again, this is because of the specific adaptations that are required in a female pelvis to facilitate labor and delivery. A note, as such, we don't do sex estimation, generally speaking, on infant and child remains because they have not undergone puberty and have not yet developed these secondary sexual characteristics, plus the skeleton itself is still in the process of developing. So as such, a lot of features on the bones are simply underdeveloped. They're not as highly visible as they are in adult skeletons. Age estimation is really critical. A number of different methods we can use that are based on generally the principle that our skeleton does change as we age, and that these changes occur within a particular time frame, right? So you think about teeth, for example. This is the most common form of age estimation that we can do, and it's based on the known pattern or sequence of eruption of baby teeth or deciduous teeth, then those tooth being lost and then replaced by adult or permanent teeth. And then we can look at wear over adulthood in order to gauge the approximate age of this individual. We can also look at growth in the long bones themselves and look at what's called epiphyseal fusion. Talked about this briefly relating to zooarchaeology, and the same principles apply, that our long bones develop at a particular rate and fuse together. at critical ages. So we can look at this. We can also look at the roughness of particular joints or ends in our bones, particularly those between cartilage and bone, because in highly mobile areas such as our rib cage or our pubic symphysis, which actually our pubic symphysis isn't that mobile. But anyways, the point is that when bones move, we can see alteration, and sometimes these alterations are indicative of age. And we can also do microstructure analysis as well. We can do some other estimations. We can estimate the stature of an individual. So here's an example of how we can take a femur and we can take some measurements. We can put it into a specific equation, and based on our knowledge of that individual's population, we can get an estimate for how tall they were in life. We can also look at, again, this idea of which population individuals belong to, and this is because populations that breed with each other have shared traits, and some traits or some populations can accumulate these traits that can be visible on the human skeleton. So for example, many European populations have high frequencies of what's called Carabelli's cusp, which is an additional cusp that forms on the molars. Other populations can see increased rates of sacralization of the fifth lumbar vertebrae. Some populations modify the body in a way that signals their group identity. So artificial cranial deformation or cranial modification. So this is an example of an individual from Peru who's had head binding. And so this impacted the shape and size of their cranial vault. Sorry, it's going a little too fast here. We could also look at individual diet and health using human remains. And if we have a number of remains from a population, we can then extrapolate this to understand what overall health was like within and diet was like within a population. So we use some of the same markers that, again, we use for mammals and other faunal remains. So we can look for skeletal markers, so things like enamel hypoplasia. These are basically lines that form in the enamel of teeth as a result of periods of feasting and famine. You can get Harris lines in bone that show the same thing. This is a sign of an infection in bone. Various forms of dietary deficiency can also impact bones. This is a form of what's called cribborbitalia, which can cause from, I believe it's iron deficiencies. Other things like syphilis can cause really extreme modifications to bone as an indicator of that disease being present. Scurvy, vitamin D deficiency, etc., all these different things. Of course, we can examine outside of skeletal markers, things like stomach contents, fecal matter, etc. Provided you an example here of how we can look at isotopes that are present in bone to figure out basically trophic levels, who is feeding on who. And what's interesting about this case study is that we see that that in this case dogs are actually being bred for food as well, that they're being potentially consumed by the humans. You can see that humans are overlapping with other sources of food, and so we get an understanding of what is going on in this particular area in terms of diet. Physical activity. Some physical activities can leave modifiers on skeletons. So on the right-hand side, we have an example of how someone being on their knees in this particular position with their toes curled under will show signs of this repetitive movement of, in this case, processing grains into flour. So doing activity with a grindstone that causes changes to the knee, to the toe joints, and of course to the spinal column as well. You can imagine carrying large loads on the head would have an impact on the bones and the vertebrae of the neck. And of course, other activities such as horseback riding, archery, a lot of other repetitive actions can also cause changes on our skeleton as well. Same with violence. We don't find a lot of evidence for violence in the past, at least not tons and tons, suggesting that people were more violent in the past than they are of today. Sometimes it's hard to determine what the frequency is like, because it's hard to determine, whether or not it was accidental or not. So here's an example of with the ancient one who had a stone tool embedded in their hip. And so the question was this an accident, or was this intentional violence against this individual? So to summarize it, we have a number of methods that we can use to analyze human skeletal remains as found in archaeological context, but I would argue that ethics has to be center and at the forefront of this kind of work.